Hi everyone, welcome back to Studying with Zahra. Today we're going to be doing Aqa Biology, and specifically we're going to be focusing on proteins. So proteins are really cool, and they're a little bit different than what we've done previously, which is like carbohydrates and lipids. And if you haven't checked out those videos, then please feel free to. I have 4 videos on them in total, and so with this we're going to end the essentially like the biological molecule part, and then we're gonna go into like the enzyme actions and stuff, which is also really fun, and it's a little bit easier, but yeah, let's get into it. So proteins are very large molecules, and the types of carbohydrates and lipids in all organisms are relatively few, and they are very similar. Each organism has numerous proteins that differ from species to species, and the shape of any one type of protein molecule differs from that of all other types of proteins. So yeah, and those are our little learning objectives. You can go through them if you like, but now we're going to go into the structure of an amino acid. So amino acids are a monomer, and this differs from lipids because we know that for lipids they don't really have monomers, and lipids aren't a polymer. But for proteins, we actually do have a monomer and a polymer. So the polymer is known as the polypeptide, not the protein. The protein is multiple polypeptides joined together. And yeah, we have about 100 amino acids, 20 naturally occurring. So you do need to know the structure of a protein. This is a very important, very common exam style question. And I'm just kind of going to be going over it briefly. So I have drawn it right here and tried to make it a little bit cuter and a bit more colorful, and therefore maybe understandable. So right here we have, let me just grab this pen. So right here is our, I hope it doesn't select it. Yeah, so right here we have our amine group. And that is basically NH2, so it's essentially just 1 nitrogen and 2 hydrogens, and it is a basic group from which the amino part of amino acid is derived. That's why it's called amine. I think I just moved that, my bad. Okay, I'm just gonna use the highlighter. I feel like it's better. So that's our little amine group. And then right here we have, at the end, we have our carboxyl group, acid. I don't know why I wrote acid, but it's acid. And we've learned about this in chemistry, but it's basically C double bond OOH. And then we have our hydrogen atom, which is right here. And then we have our R group. So that's our R group. And the R group over here looks like this. And over here, that's your amine group, that's your hydrogen, and then that's your carboxyl group. Now, once again, you do need to know the structure. It is so important, and it's such a common exam question. So once again, this is right here is our carboxyl group with C double bond OOH. Next, you have your amine group, NH2, just a small little hydrogen atom, which is just H, and then our R group, which is C, and then R is just variable depending on the protein. And so yeah, so the formation of a peptide bond, and then we have the primary structure. Proteins are a little bit longer and they're a little bit trickier, but it's okay, we will get through them in this video. So yeah, let's get into it. So the formation of a peptide bond. In a similar way that monosaccharide monomers combine to form disaccharides, amino acid monomers can combine to form a dipeptide. So that's just 2 amino acids. And disaccharides are basically 2 monosaccharides, so that would be for carbohydrates. An example might be, I don't know, lactose, I suppose. And the process is essentially the same. So you have the removal of a water molecule in a condensation reaction. We know that a condensation reaction is, sorry guys, is when a bond forms and a water molecule is released, so H2O. So we have an OH removed from the carboxyl group and an H from the amino group of another amino acid. So if I was to kind of draw a bond or present a condensation reaction between these two amino acids that I've drawn right here, this one and this one, I would take the OH from here, and then I would take the hydrogen from the amino group, and you need to do it from the amino group. So I'll be right there. And then, or it could be vice versa. So it could be like the OH from here and then the H from here. Either way works. But yeah, that's just the condensation reaction. That's going to form a peptide bond. So unlike carbohydrates where we had a glycosidic bond, here we have the name is called a peptide bond between the carbon atom and the nitrogen atom. So it would essentially be between here and here. Right there. Perfect. So the formation of a peptide bond is illustrated in figure 3 in a similar way as the glycosidic bond of a disaccharide can be broken by the addition of water. So the peptide bond of a dipeptide can also be broken by hydrolysis to give its two constituent amino acids. Okay, so the primary structure of proteins. So through a series of condensation reactions, many amino acid monomers can be joined together in a process called polymerization, and the resulting chain of many hundreds of amino acids is called a polypeptide. The sequence of amino acids in a polypeptide chain forms the primary structure of any protein, as we shall see in topic 8.1. So over here, that's my little peptide bond. You can see my C double bond that used to have an OH and that used to have the H. So that's how we kind of derived the H2O from there. And now we have a peptide bond right there. The yellow, I think that I've kind of highlighted in yellow, is my peptide bond. And then contrastingly, I have my OH and my H that forms that, and a bond is going to form between the C and the N that is called the peptide bond. Very, very cool. Okay, so they've kind of drawn it here. Now, polypeptides, yeah, so the primary structure of proteins is essentially just amino acids. So once a bond is formed, and I've made a couple of notes in the margins, once a bond is formed between adjacent amino acids, the amino acid is folded up to give a 3D protein structure, and this has 4 stages. It has primary, secondary, tertiary, and quaternary. So essentially this might seem a bit overwhelming, but once you've had your bond formed between your 2 amino acids that are next to each other, the amino acid, now it should be amino acid chain, or now your polypeptide chain might fold up to give a 3D protein structure, and that's how a protein is actually formed. So the primary structure determines its ultimate shape and hence its function. And the way that it's folded the way that the sequence is can really impact the function of the protein significantly. So yeah, so we're gonna start with the secondary structure. So the linked amino acid groups make up the polypeptide possess both the NH and the C double bond O groups on either side. The hydrogen of the NH group has an overall positive charge, while the oxygen has a negative charge, so this forms a weak bond known as a hydrogen bond. And this causes the polypeptide chain to be twisted into a 3D shape, such as a coil known as an alpha helix. And this is kind of your structure of the alpha helix. So an alpha helix can be further twisted to form a tertiary structure. So once again, for our secondary structure, we have our H on the NH group, and that has a slight positive charge, and then we have our O on the OH group, on the CO group, that has a slight negative charge. This forms a weak bond known as a hydrogen bond, and this causes the chain to be twisted into a coil known as an alpha helix, and this is what our alpha helix looks like. So once again, my CO has my oxygen, slightly negative, NH has hydrogen, slightly positive, the two bond together, a weak bond is formed. This results in the coiling and folding of the structure, resulting in an alpha helix formation. For the tertiary structure, the alpha helixes, I don't know why they wrote helices, I'm pretty sure that was like a typo, but the alpha helixes can be twisted and folded even more to give the complex and often specific 3D structure of each protein. This is known as the tertiary structure. Now, this structure is maintained by a number of different bonds. Where the bonds occur depends on the primary structure of the protein. So we have our disulfide bridges, which are fairly strong and therefore not easily broken. And then we have our ionic bonds, which are formed between any carboxyl and amino acid groups that are not involved in forming peptide bonds. They're weaker than disulfide bonds and they're easily broken. And then we have our hydrogen bonds. Now, once again, this might seem a little bit overwhelming, but I am going to go over it right now. Now, if you need a second to just read it, then feel free to just take a second and read it, because in order to kind of understand it, it is very important. Okay, so the alpha helixes can be twisted and folded even more to give a specific 3D structure. And where the bonds occur depends on the primary structure of the protein. So the bonds maintain the structure. We have our disulfide bridges, which are fairly strong. They're not easily broken. So these are formed between our sulfurs. Ionic bonds, which are formed between any carboxyl and amino groups not involved in forming peptide bonds. They are weaker than disulfide bonds and easily broken by changes in pH, which is why pH is so important in terms of essentially maintaining all of life, really. And then hydrogen bonds, which are many in number but very easily broken. So our alpha helixes can be folded again and again into complex shapes to create a specific 3D structure known as the tertiary structure. And the tertiary structure has certain bonds that maintain the structure. These bonds are dependent on the primary structure, and we have three different types of bonds, namely disulfide bonds, which are very strong, disulfide bridges, which are very strong, not easily broken, hydrogen bonds, which are very weak but many in number, and then last but not least, we have our bonds between our amino groups and our carboxyl groups that are not involved in the peptide bonds, and this is known as an ionic bond. These are more easily broken than the disulfide, and they're easily broken by changes in pH. So ionic bonds are formed between the amino group and the carboxyl groups that are not involved in the peptide. The 3D shape is really important because it makes the protein distinctive. So that's my primary structure, my secondary structure, hydrogen bonding between the O negative, slightly negative O, and the slightly positive H, tertiary structure, and then we have our quaternary structure. This is a little bit trickier, but not too bad. So large proteins, often in the form of complex molecules, containing a number of individual polypeptide chains that are linked in various ways. So they may be non-protein, and such as the iron-containing heme group. 3D structure is important. The sequence of amino acids determines the 3D shape in the first place. So the quaternary structure arises from the combination of a number of different polypeptide chains and associated non-protein groups into a large complex protein molecule. So once you have your tertiary chain, right, you have your polypeptide chain, and multiple polypeptide chains may combine, and you might have non-protein groups that may be added that results in the formation of a complex protein molecule. Test for protein is really easy. You add burette, and it should go from blue to kind of a purple. And if it stays blue, then, well. The disulfide bond is a covalent bond. And then we have our bond types. So hydrogen bonds are the weak bonds between the positive charge on the H and the negative charge of the O. Disulfide bonds are covalent bonds between and then we have ionic bonds, which are formed between the positive and negative amino acid side chains. I'm not sure how accurate that is, actually. They're formed between the carboxyl and the amino acid. Then we have our summary questions. Totally try them out. And then we have our protein. This is just kind of extra stuff. But the 3D shape of protein is maintained by bonds. Hydrogen bonds. Hemoglobin is a globular protein that has 4 polypeptide chains joined by disulfide bonds. Each chain is arranged around a heme group that contains an iron group that binds with oxygen. That's pretty much it for proteins. It was a little bit difficult, and if you guys want me to film another video on this, please feel free to let me know. But that was pretty much it. Again, it is a bit difficult, but don't be afraid. And yeah, thank you so much for watching.